Hello everyone, and thank you for joining us for our Cell Press webinar, The Role of Genomics of Biodiversity Conservation in a Changing Climate. I'm Andrea Stevens. I'm the Editor-in-Chief of Trends in Ecology and Evolution, and I'll be your moderator for today's webinar. Climate change represents a threat to many species and populations. Conserving these populations requires us to understand how plant and animal populations might adapt to novel conditions. In this webinar, we aim to highlight how genomic technologies can be used to tell us about the evolutionary history of Earth species and how we can best preserve these species for the future. We've got three speakers today. First up, we have Sally Aitken, who's in the Faculty of Forestry and the Biodiversity Research Centre at the University of British Columbia in Vancouver, Canada. Sally will discuss local adaptations to climate and boreal tree populations. Next up, we have Comeault from Bangor University in Wales, the UK. He will describe how he uses fruit flies to investigate how populations evolve under different conditions. And finally, we have Orly Razgour from Exeter University, also here in the United Kingdom. She focuses on the role of plasticity versus a genetic adaptation in species response to climate change. She does this work mostly in bats. Before we get started with the talks, we have a few practical points. All of our speakers will talk for approximately 15 minutes. Following the talks, we'll have a Q&A session which covers all the three presentations. If you have a question for any of the speakers, can you pop it in the Ask a Question box below the presentation screen? Just type it in here. You can enter this question at any time. You don't need to wait till the end. We will do our best to pass on as many of the questions to the speakers as we can during the Q&A. It's really helpful, though, if you include the name of the speaker to whom you would like to address your question, although other speakers may jump in as well. And before we begin, we'd just like to thank our speakers for making the time to join us today, and also our sponsor, Illumina, for supporting the webinar. And now, kicking off the webinar, it's our first speaker, Dr. Sally Aitken. Sally, thank you for joining us today, and take it away.
Thanks very much, and thanks for the invitation to speak today. I'll just pull my slides up. So I am speaking to you from the University of British Columbia on the traditional, ancestral, and unceded territory of the Musqueam Nation. And I'll be talking about genomics of climate adaptation in conifers. So in conifers, we see widespread species have moderate to strong local adaptation to climate. Here we have trees that have been grown in a common garden that originated from the locations written above them with a wide range in mean annual temperatures. Trees from warm environments grow larger because they grow for a very long period. Trees from colder environments are smaller, but they are more cold hardy. And we see these kinds of trade-offs across many species. Now, the problem for trees is that the rate of climate change per generation is very high because of their longevity and their time to sexual reproduction. So we've got populations adapted to historic climates, and as local climates change, those populations become mismatched with future climates. And we've already seen tree mortality increasing with climate change, especially in Western North America. So what do we need to know to understand and manage the risks of climate change to populations? And in the case of trees, we want to inform assisted migration within and sometimes outside of species ranges to assist the shift of genetics and populations as climates change. So we need to know the strength of local adaptation. We need to know what drives local adaptation, especially what aspect or what variables of climate drive that local adaptation. We need to have projections of how species climatic niches are going to shift geographically. And we want to be able to identify those populations that are at the greatest risk of maladaptation or extirpation. Now, we may or may not need to know what genes are involved in climate adaptation. So I'm going to break this talk into two parts. The first is lessons from lodgepole pine, Pinus contorta, which is a very widespread species in Western North America. And we studied over 280 populations from Western Canada of this species. We did exome capture and then analyzed genotype environment associations and did genome-wide association studies of traits to identify candidate SNPs involved in climate adaptation. And there are about 32,000 of those on a SNP array. We also had phenotypic data. We grew all these populations in common gardens, and we phenotyped traits like growth, timing of growth, initiation and cessation, and cold hardiness. We also have an independent data set for a reciprocal transplant field experiment with trees that had 20-year data. So over on the right here, you can see a map of the results of the cold hardiness assays from artificial freeze tests. The populations that are in blue had very little cold injury in those tests. The populations that are red had very high levels of cold injury. And you see a geographic pattern here that corresponds to extreme low temperatures and mean winter temperatures. And this is a typical pattern that we see where cold adaptation to low temperatures has the strongest signal of local adaptation. So one of the important questions we've asked with genomic data is, can we identify the same climate variables as driving local adaptation? Because if we are going to manage populations for a changing climate, if we are going to use assisted migration, we need to know what climate aspects are most important. And so on the x-axis here, we have the climatic variance explained by the 20-year field common garden experiment, and the y-axis is the climatic variance explained by genomic data. And in fact, both of those data sets identify the same set of low temperature variables as being most important. But we do have a signal that summer moisture is important in the genomic data, and that perhaps reflects extreme events. Now, a very nice review paper by Thibaut Capelon and others reviewed the steps for really assessing potential future maladaptation of populations. And the first two steps are to generate genomic data and then do genome scans on those data. And so this is with the goal of identifying markers that are associated with climates or climate relevant traits, as we have done. And the next step is to develop predictive models that, first of all, are based on the turnover of adaptive alleles along climatic gradients, where we have some variation in allele frequency for adaptive markers along some environmental gradient. And one approach to doing that is to use a program called Gradient Forest, which is an extension of random forest. It's a machine learning regression tree approach to characterize this turnover of alleles. So one of the questions we asked with lodgepole pine is, do we see the same patterns in genomic data that we see in phenotypic data, or do we see localized genomic declines? And so here we have clusters, 6 clusters of SNPs that were associated with cold injury. We find many SNPs associated with cold injury, and you can see 6 different patterns here. And for a moment, I'd just like to focus on this cluster 4, where we see little change in allele frequency at very cold temperatures in terms of mean annual temperature, but a steep climb at warm temperatures. And that's versus cluster 6 here, where we see variation at cold temperatures, but not at warm temperatures. If we map the distribution of variation in those 2 clusters, what we see is a marked divide corresponding to the Rocky Mountains running here between British Columbia and Alberta. Cluster 4 has lots of variation in warmer populations, but very little variation in cooler populations. And we see the reverse for cluster 6. The good news from this analysis is that almost every population was polymorphic for almost every SNP that was associated with this trait. So following up on that with the same data set, UAU looked at she used gradient forests to cluster similarly adapted populations of lodgepole pine. So here we see a map using an analysis of the full SNP data set, nearly 30,000 SNPs, and each color represents a cluster that are similarly adapted trees. And we see the top climate variables here relating, the first three relating to temperature and the fourth relating to evapotranspiration. When she ran this on a candidate set of just under 1,000 SNPs from GWAS, we see essentially the same map, and we see very similar importance values for climate variables. So this is good news, suggesting that we don't need candidate sets to do these kinds of analyses. So the next steps identified by the Cat Blanc and others here in these figures are to, we use the genetic environment relationship and the model from, for example, gradient forests to project ahead what the offset is going to be between the current genetic composition and future climate. And after doing that, we really need to validate projections with an independent data set. So now I'm going to talk about a very recent analysis with Douglas fir. This is a species that also occurs along the Pacific coast, and we have two varieties. We've got coastal Douglas fir in the green shading and interior Douglas fir in the purple shading. So we had 74 populations, did pooled exome sequence capture, and got about 10 million SNPs. These are huge genomes, about 20 gigabases, so we generally target the exome. And then Brandon Lind led this work and projected the offset, again, using gradient forests. And we asked the question, do genetic offset projections match phenotypic data from the Common Garden experiments established by Rafael Candido Ribeiro? Now, the Common Garden experiment is here in Vancouver, and these trees were brought from nearly all across the range to Vancouver, but note that the Common Garden is in the range of the coastal variety. So here are the results of an analysis showing the validation score based on the correlation between the projected maladaptation, the projected offset, and growth in the Common Garden experiment. We would expect a negative relationship, and in fact, we see a strong negative relationship there based on the distance, the climate distance that these trees have been moved to the location of the Common Garden. So this says that gradient forest models, model prediction of offset are validated by the phenotypes. What I haven't shown here, but these gradient forest models based on genomic data outperform just using climatic distance measures or geographic distance to estimate offset. And Brandon also found that random SNPs here performed fairly equivalently to candidate SNPs based on genotype environment association. So that's more good news about random SNPs. However, when Brandon did the analyses separating the two varieties, first of all, we see that the validation scores were inflated, especially for the interior variety compared to analyzing across the species. But perhaps more important than that is if we look at maps of projected offset, here darker blue colors project less maladaptation in the future. The red shades project high maladaptation in the future. And if we compare the map for both varieties with the map for the interior variety, and we can zero in, for example, on this area, and we see that the model for both varieties combined is projecting high maladaptation for these populations in the sort of interior Pacific Northwest. But the model for the interior variety is actually projecting low maladaptation for the same populations. So we still got to work out some things about how these models work and what the appropriate scope is for analysis. But these methods are very much under development by many people right now and under validation, and we're learning more all the time. So in closing, genotype environment associations can be very useful to identify the climatic drivers of local adaptation. And this can inform assisted migration and other management actions for populations and species of concern. Random SNP data sets for these analyses I've showed you perform as well as candidate sets for projecting maladaptation. But of course, candidate loci provide many biological insights that we don't get from random SNP sets, for example, from RAD-seq kinds of approaches. Range-wide analyses across subspecies or varieties may inflate relationships, so we have to be really careful about extrapolating models outside the scope of samples. In our case, we saw those results that weren't well validated when we extended the model to areas that we didn't, that weren't typical of the common garden, and other issues there with population structure. And in closing, maybe a more philosophical question, but I would ask if, based on what we know now, should we prioritize individual populations for climate-related management, given the uncertainties of offset approaches and uncertainties around climate trajectories, or should we be managing all populations for climate change until we have better information? With that, I would like to thank in particular all of these fine people who really generated the data and did the analyses I've been talking about, and our many sponsors of this work for AdaptTree and co-AdaptTree projects, especially Genome Canada, Genome British Columbia, and the other genome centers. Thank you very much.
Thank you very much, Sally. That was a great talk. Now we're going to move on to Aaron Comeault from Bangor University. Aaron, thank you very much for being with us, and do you want to take it away? Aaron?
Yes, sorry, I was waiting for the box to give me the option to share my screen. Sorry. Start sharing. All right, thanks very much for the organizers for the invitation to present. I'm very excited to present some work we've been doing testing thermal adaptation during invasions in the African fig fly, Zapyronus indianus. And so just to give my talk a little bit of context, when I think about the conservation genetics umbrella, frequently we think of these species of conservation concern. They may have small effective population sizes, potentially suffering from inbreeding. We're also really interested in metapopulation dynamics and how adaptation might play out under different scenarios, as Sally was just talking about. But I'd like to also argue that introduced species maybe have a place to sneak under the broader conservation genetics umbrella for a number of different reasons. But one of those is if we think about the factors that are affecting species of conservation concern, such as exploitation, habitat fragmentation, pollution, disease, and climate change, well, introduced species also cope with these factors and may be good models to understand how species can respond under changing environments. A nice example of this, a paper published in Science in 2013 by Colati and Barrett, where they looked at the introduced species purple loosestrife in North America along a latitudinal cline in northern, central, and southern populations. And they found evidence for local adaptation where, when grown under common garden environments, the local population would have an advantage. So if you had individuals collected from a population in a northern environment and you raise them in the north, they have higher fitness than individuals collected from southern populations. And they identified a suite of traits that kind of affect this fitness in the different populations. And so an example of how an invasive species might be used to identify traits that can be informative for understanding adaptation more broadly in other species as well. Introductions are also really interesting to me personally, at least, because they're very dynamic and they're at non-equilibrium kind of states. And so you can think about invasions or introductions as occurring across geographic and temporal kind of continuum, where you have initial dispersal and colonization of a novel environment and subsequent expansion into a new range. During this colonization and expansion process, species are going to be exposed to novel environments and may or may not adapt to those environments. And so we've been studying invasion dynamics in this species here, the African fig fly, Zaprionis indianus. This species is native to sub-Saharan Africa, and it's now broadly distributed across sub-Saharan Africa. But it's undergone a rather dramatic and nearly cosmopolitan range expansion, first being found in the Indian subcontinent, and more recently in the Western Hemisphere, showing up in South America and North America. In South America, it was first reported in Brazil, but it subsequently spread to be broadly distributed across South America. And in North America, it was first reported, or at least in the US, sorry, it was first reported in Florida. And since it's expanded its range, it can be found as far north as southern Canada. The species utilizes a broad range of host fruits where it will find typically damaged or rotting fruit. It'll lay its eggs on the fruit where the larvae will then develop in the fruit and eclose into adult flies. And it is considered a pest species on some important crops in some areas of its distribution. And so when starting to study this species, we wanted to ask the question, well, given this broad distribution and the different thermal environments that they might be, the individuals are exposed to, what can we say about evolution of thermal performance, or more specifically, do thermal traits undergo rapid local adaptation in this species? And so to get at this question, we've been relying primarily on thermal performance curves to estimate proxies for fitness. And so how these kind of measurements go is we have a vial that contains a medium where the flies will lay their eggs and their larvae will develop. And we place two flies who have not yet mated in a vial. We allow them to mate for some set period of time. We then clear the adults out of the vial so the eggs will remain in the vial. And then we allow the eggs to develop and the larvae to eclose into adult flies and count the total number of flies that emerge from this vial. And so it's kind of a one generation measure of fitness. We do this for multiple pairs, randomly allocated to one of 5 different temperature treatments, ranging from 14 degrees to 34 degrees Celsius. Using these data, we then use a hierarchical Bayesian model that was developed by Silas Teitz and colleagues and published in 2019 in the American Naturalist. And this allows us to fit thermal performance curves for every population or species that we collect these measurements from. And the nice thing about the thermal performance curves is that hyperparameters you can kind of extract from the curves provide useful information on a species or population's thermal biology. So I'm going to talk about 3 of those: thermal niche breadth, or B50, thermal optimum, the optimal temperature at which fitness is kind of maximized, and T-min, or thermal minimum, and that's the estimated temperature where fitness decreases to 0. And so we've been measuring these thermal performance curves for a number of different populations and species, but I'm just going to talk about different populations of Zaprionis indianus today. And so we've looked at thermal performance in 3 populations in the introduced range in North America and 3 in the native range in Africa. And so what did the results, what did the data look like? When we look at thermal niche breadth, the majority of populations have a similar thermal niche breadth, whereas there's this one population we sampled in New York that looks to have a significantly broader thermal niche than the other 2 populations in the introduced part of the species range, but it doesn't significantly differ from the populations found in the native part of the species range. Looking at thermal optima, we find a very similar pattern, really, where this New York population kind of pops out and looks like it has actually a higher thermal optima than all the other populations we looked at. Again, it didn't significantly differ from the two populations, or from two of the populations we sampled in Africa. And I think most interesting for me is when we first set out to kind of study these populations, the hope, I guess, was, or the prediction was, that these individuals collected from more northern latitudes, so in New York, in North America, might have adapted to cope with colder temperatures than the populations found in warmer, more southern climates. But that wasn't the case. We didn't find any evidence that the New York population or North Carolina, the more northern population, had adapted to cope with lower temperatures. And so, at least based on these lab-based thermal performance curve estimates of performance, we found no strong evidence for adaptation to cold temperatures in the northern population. And thermal performance curves, or aspects of the curves, are largely shared across populations in both the introduced and native parts of the species range. But what we wanted to do next was look at this under more natural conditions. So we conducted a small common garden experiment where we set up population cages, 3 with individuals founded from North Carolina and 4 with individuals founded with individuals from Florida. This was run in North Carolina. So North Carolina is the local genotype or local population. We then sampled the cages every 2 weeks for reproductive activity. And how we measured reproductive activity was we put in vials that were fresh food vials. We left them in the cages for 24 hours and then went back and counted the total number of eggs laid in those vials as a measure of reproductive activity. And what we found was that reproductive activity didn't significantly differ across all our sampling times between the different populations. And there wasn't a local adaptation. So remember, the black curve here is actually the local population in this study. And more interesting, I think, is that on the November 16th date, you see that reproductive activity fell to 0 and it didn't recover. And actually, by the middle of December, all the adult flies were dead in the cage as well. If we overlay temperature, this is measured hourly, I believe, throughout the course of the experiment, when this reproductive activity drops to 0 is kind of coincides with when temperatures, evening temperatures or nighttime temperatures, drop below 0, suggesting that there's this lack of ability to cope with cold temperatures, even at the local population that was sampled from North Carolina. We also did community sampling throughout the year just to see how abundant this species was throughout the year. And so what you're looking at here, each circle represents a sample of Drosophilid flies, sampled from my compost pile. And the red dots are the number of individuals from that sample that were Zaprionis indianus. And you can see their abundance or membership of the Drosophilid community is concentrated to kind of late August into October. And so, despite the temperatures throughout the year being conducive to allowing the species to be reproductively active, we don't find them until later on in the year. So taken together, information we have from thermal performance curves in the lab, this common garden experiment, and then abundance throughout the year provides relatively weak evidence that there's been adaptation to cope with cold temperatures. And it led us to come up with a hypothesis that maybe these northern populations aren't actually resident established populations, but rather they're each year colonizing northern environments from their kind of resident southern populations. And that's what generates this lag between suitable climatic conditions in northern sites or northern locations and the time when we actually find Zaprionis indianus in the Drosophilid community. And so how can genomics maybe help us get at these hypotheses? I'll present some very preliminary data and kind of designed that I think could help us get at understanding whether there is this kind of yearly colonization of northern latitudes or actually established populations, so using patterns of genetic variation. And so using a kind of a simple prediction is if we have yearly colonization of northern sites from southern locations, then allele frequency turnover between years at northern locations should be relatively high, especially if there's kind of bottlenecks and colonizations every year happening as the species moves up to northern environments. Secondly, differentiation between locations within a given year might be low because the same kind of genetic variants are making their way up to those northern populations every year. And so there's not local adaptation and differentiation building up between populations at different locations. So thinking about correlations of allele frequencies between populations, imagine we have a sample from North Carolina in 2 years. Between years, we might expect that correlation to be weak if there's this serial colonization every year. But within a year, there might be a strong correlation because the same genotypes have successfully colonized those northern environments. And the kind of reverse might be true if there was not serial colonization every year. And so we collected a small genomic data set to kind of try to start to look at this and look at correlations in allele frequencies between populations between years. So first, looking at the North Carolina population sampled in 2 years, we find allele frequencies are highly correlated even between years. So remember, this would go kind of against the simple prediction we made about allele frequencies being correlated, not being correlated if there was serial colonization every year. We also found really strong correlation between allele frequencies across locations, so between the North Carolina population and the New York population within a given year. So this is kind of consistent with our naive prediction that we might expect if there was serial colonization each year. We also, so both patterns are showing that allele frequencies are just highly correlated, both between years, at least in this data set, and across locations. We also collected some data from another Drosophilid that's also an introduced species that has, roughly speaking, similar time frame of colonization of these environments. And between the northern and central population, we find a weaker correlation between allele frequencies. And it's interesting because this species, Drosophilus suzukii, is much more cold hardy than Zaprionis indianus when we test them in the lab and whatnot. So one interpretation of these patterns is that there's simply a single large panmictic population of Zaprionis indianus that might be colonizing northern environments each year, but that colonization isn't associated with kind of a bottleneck and a loss of genetic diversity and a structuring of genetic diversity between years. But we need more data to test that. And so going forward, I think using these kind of broad temporal and also geographic sampling designs can provide important insights into the dynamics of adaptation, particularly in these non-equilibrium systems that are maybe colonizing new environments relatively recently. And I've presented results from a limited number of thermal performance traits, but building up additional data sets where we test differentiation in other traits that might be important for survival in different environments will, again, help provide additional insight. So combining genomic and phenotypic data can help to identify traits and genes that might be important for adaptation to these different climates. And with that, I'd like to thank you for listening. Thank you again for the opportunity to present, and I'll acknowledge all of my important collaborators who have contributed invaluably to this work. Computational support from the University of North Carolina Research Computing and Supercomputing Wales here in the UK. And then funding from Bangor University and the NSF. And thanks very much.
Thank you, Aaron. That was really interesting. Remember, everyone, if you've got any questions for Aaron or Sally, could you please type them into the chat box below? We've got some questions coming in, but I'm sure you've got plenty of others. And now, last but not least, we have Orly Razgour. Orly, thank you for joining us today, and take it away.
Thank you. I've got my Pokeballs. So I'm just going to be sharing my screen. Okay, so today I'm going to be talking about how we can use integrated genomic approaches to investigate responses of biodiversity to climate change. And I'm going to be focusing on bats. So if you're joining this symposium, this webinar, I'm sure you agree with me that climate change is a major threat to global biodiversity. Not just biodiversity, but also the ecosystem services they provide, which is something that affects our food security for us and our survival in general. So several IPCC reports have come out and shown how the climate is already changing. And this climate is further projected to change over the next few decades. So temperatures are projected to increase, patterns of precipitation will become a lot more variable. Now, this is something that's already affecting species and resulting in many organisms shifting their ranges in pursuit of more suitable conditions. However, climate change is not happening it's happening over a backdrop of extensive habitat loss, degradation, and fragmentation. And this is something that's already resulting in biodiversity declines, but will also affect the ability of species to shift their range in pursuit of suitable conditions. So how biodiversity is going to respond to these interacting stresses is something that really will depend on the extent of change, but also on intrinsic factors. So this is the ability of species to respond to these changes. So their sensitivity to the changes, their adaptive capacity, and their ability to move away. So commonly, biodiversity vulnerability to climate change is assessed through using correlative climate niche models, also known as species distribution models. And these models relate information of where species are currently found to climatic conditions experienced by the species. And these models can be used to project where future suitable conditions for the species will occur. Now, these models are widely used. However, they've been criticized for being oversimplistic and disregarding important mechanisms that affect species' responses to climate change, like evolutionary mechanisms, adaptive capacity, and dispersal. So the research carried out in my group uses integrated genomic approaches to address some of these gaps in our understanding of biodiversity responses to climate change. So what can genomic data actually tell us? Well, first, it can tell us something about adaptive responses to climate. And we've seen Sally's talk that really showed how we can use that. Second, genomic data can be used to identify landscape barriers to gene flow. And as a result of that, can tell us something about barriers to range shifts. Genomic data can also tell us something about population status and vulnerability. And finally, we can use genomic data to try and disentangle the role of phenotypic plasticity versus genetic adaptations. I'm going to touch on all these issues in this talk very briefly. So first, let's start by looking at how we can use genomic data to identify adaptive responses to climate. So we developed a framework that helps us to directly integrate and consider adaptive genetic variation in response to climate into our vulnerability assessments. And I'm going to show you how we can implement this framework through an example of two Mediterranean bat species, Myotis escaleri and Myotis crypticus. So we're generating genomic data sets for these bats. Then we use these genomic data sets to try and identify climate-adapted individuals. So we use genotype environmental association analysis, something that's already been mentioned in previous talks, to identify genomic regions that are associated with climatic conditions likely to change under climate change, and likely to also be important for their reproductive success and survival of the species. So in the case of these bat species, these are maximum temperatures and summer rainfall. Then what we did, we plotted the individual bats across the ordination space based on where they fall relative to those two environmental conditions, based on their genetic makeup in those genomic regions that are associated with these climatic conditions. And we divide our individuals into those that are associated with hot and dry conditions, here in white, versus those that are associated with cold and wet conditions in grey. And these are the individual distribution for the two bat species. Then we directly incorporate this information into our models. So the models that you see here on your left were generated using all the location records of the species. These are traditionally used species distribution models. What we see here is the distribution or the spread of climatic suitability for these bats, from low suitability in blue to high suitability in orange. The map at the top is under present condition, at the bottom projections for the end of the century, future conditions. And if we compare the two maps, what we can see is projected extensive range losses and loss of suitable conditions around the Iberian Peninsula, which is the main place where these bats are found. Now, what we did, we generated separate models based on their genomic information for those individuals that are adapted to hot and dry conditions in red versus those individuals that are adapted to cold and wet conditions in blue. And when we did that, if we compare these two maps at the bottom for future conditions, what we can see is that when we consider adaptive variation, we have reduced range loss projections in our models. So next time I'm going to move into how you can use genomic approaches to identify landscape barriers to gene flow and range shifts. And we're going to move to a different bat species, the grey long-eared bat, Plecotus austriacus. So what we're doing here, we're using the landscape genetic approach to relate patterns of genetic variation to landscape heterogeneity. So in the case of this bat, we compare different landscape variables that may be affecting gene flow between our studied colonies. And what we found that the main landscape variables that affect genetic connectivity is landscape resistance as a result of habitat suitability, which is the outputs of our species distribution models. So then we took this information to try and understand how it will affect future range shifts. So here we have maps of potential movement density based on the effect of the landscape barrier on genetic connectivity. We can see that under present conditions, all our colonies, in particular around the Iberian Peninsula, are nicely linked together. So yellow colors represent high movement density, blue represents low movement density. Then we projected to the future. What we can see that under future conditions, several of the populations or colonies we sampled are likely to become isolated and unable to shift the range as a result of the effect of landscape connectivity and changes in landscape connectivity. So we also use this approach to look at the effect of landscape barriers on evolutionary rescue potential. And by evolutionary rescue potential, what I'm talking about here is the potential of gene flow from populations that are adapted to hot and dry conditions into maladapted populations. So these are populations that are adapted to cold and wet conditions. So we're coming back to our two Mediterranean Myotis bat species here. Using the landscape genetics approach, we identify that genetic connectivity in these bats is primarily affected by forest variables and topography. And then we projected the potential gene flow between populations or colonies that are adapted to hot and dry conditions into populations that are adapted to cold and wet conditions, so maladapted. And what we can see here again, movement density, yellow represent high movement potential, that we do have a potential for evolutionary rescue. However, this gene flow of adaptive variation is not likely to reach all populations. Okay, next I'm going to move into talking about how we can use genomic approaches to monitor responses to environmental change. And as part of the GeoBond genetic composition group, we recently published a set of essential biodiversity variables that can be used to monitor changes and responses to environmental change in terms of genetic changes in the population. However, for most species, we don't have these kind of long-term monitoring data that will enable us to compare responses to environmental changes. So instead we can use genomic approaches to try and disentangle the demographic history and changes in population size over time. Now, the reason why we can use it is because the individual genome has got inside it information on ancestral genomes, as well as the evolutionary forces that shape them. In this study, we used approximate Bayesian computation model-based inference of demographic history to study population changes in the barbastel bat in Britain. So we compared different models of population change to a non-model of no change in population size over time. And a model that received the highest support is a model that shows a severe population decline of 99% in both the southern and northern British populations. Now, the timing of this decline is in the past few hundred years. And if we look at the history and what happened around that period, this period was a time of the early colonial expansions and colonial explorations, which necessitated very extensive shipbuilding. Now, shipbuilding was facilitated through using those very large, mature oak trees. Now, these large, mature oak trees are the same trees that are used by the barbastel bats for roosting, hence the impact of the shipbuilding on the population declines of these bats. I'm going to finish by very briefly talking about the role of phenotypic plasticity versus genetic adaptation and how we can use genomic approaches to try and disentangle that. So we're trying to do this as part of the adaptation hologenomics project, which I co-lead with Anton Alberdi from the University of Copenhagen. And this project is part of the Earth Hologenome Initiative. This project is a very wide collaboration with many collaborators across Europe that aims to generate paired animal genome and gut bacterial metagenomes to understand how small mammals in Europe are adapting to climate change. So what we're particularly interested in seeing is how the gut microbiota, which can adapt and respond much faster to environmental changes, affects adaptation and responses of the host to environmental changes. So this way we can really see whether the adaptive, the gut microbiota confers a mechanism of phenotypic responses to environmental change in the host. So some brief take-home messages. I hope I managed to convey to you the importance of using integrated approaches and combining genomic, ecological, and environmental data with different modeling approaches to understand biodiversity responses to climate change. The use of genomic approaches has shown that current models used to look at vulnerability to climate change may be overestimating range losses because they disregard the effect of local climatic adaptations. Genomic approaches also can be used to look at the impact of landscape connectivity on range shifts and evolutionary rescue. And in terms of conservation management, it can help us identify those landscape variables that we may need to work on and to manage to increase connectivity. Responses to climate change may be affected by historic population declines, and that means that it's really important to understand what is the population status currently relative to historic population sizes, and genomic approaches can really inform on this. And finally, we can use hologenomic approaches to try and disentangle the role of phenotypic plasticity versus genetic adaptation to climate change. I wanted to finish by acknowledging the different funders of my work and the many collaborators that were part of this work. And thank you for your attention.
Thank you, Orly, for the really nice presentation. Thank you to all our speakers for sharing all their discoveries about how plant and animal populations are responding to climate change and what genomics can tell us. If you've got questions, please pop them into the attendee question box. And we're just going to take a few minutes now to go over some of the questions that various listeners have submitted. First question has come from Omar Ramirez Flores. He's asked it to Sally, but I think it would be interesting to get all of the speakers' views on this question. He's really asked, what is a maladaptation? What do you consider a maladapted trait to be? Sally, do you want to start?
Sure. And thanks for that question, Omar. So in the context of trees, maladaptation would be a trait that reduced fitness under new conditions. And for an example of that, we see that these trees are adapted to the length of the growing season. When our conditions favorable for growing. If a tree starts growing too early, or it stops growing too late, it risks injury from frost. But if it grows for or drought at the end of the season, but if it grows for too short a period, it's not very competitive and doesn't grow enough to compete effectively for light. So a shift in climate that results in a trait that reduces the growth and reproductive capacity of a tree, that would be maladaptation.
Aaron, Orly, what do you define maladaptation to be in your systems?
So I can say that in the cases of many bat species, we do know that bats that are found in arid environments tend to have lower evaporative water loss rates, for example. And this is something that enables them to survive in an environment that has less water availability. Now, if we think about climate change, one of the things that are going to change in many parts, in particular around the Mediterranean basin, is reduced summer rainfall, and as a result of that, reduced water availability. So that means a maladaptation in the bat will be an inability to survive these hotter and drier conditions because they're losing too much water for evaporation through their very large and membranous wings. So this is just an example of how, in this case, droughts and reduced water availability is likely to result in maladaptation in populations that did not necessarily evolve under conditions that don't have as much rainfall.
And how about in your systems, Aaron?
Yeah, not too much to add. Very similar. With the fruit flies I was presenting about, maladaptation could be seen as the inability to cope with cold temperatures. So in the northern parts of their environment, if the hypothesis that they're not resident there and they're not able to cope with the cold temperatures, and from that perspective, they're maladapted to the cold temperatures that allow them to survive throughout the winter.
Cool. Next question, another question for Sally. Emma Yee asks, is global warming good for lodgepole pine since it provides more variation? Oh, Sally, I think you're on mute.
Not the first time that's happened. So the maps I showed of variation levels are the existing variation. Those weren't projections forward. It's true that populations with more variation should have more capacity to adapt to new conditions. But the question of generally whether global warming will be good for lodgepole pine, other analyses that we've done suggest that there are populations in very cold locations that are actually adapted better to somewhat milder conditions, somewhat warmer conditions. And so there are some populations that initially we expect to grow more with a bit of climate change, but that is going to turn around in, you know, maybe 20 or 30 years for those populations, and growth is projected to decline. And there are many other populations that are expected to decline with every bit of climate warming. So it is population dependent, but overall, the picture is not a happy one. And we see some areas that are no longer going to be suitable for lodgepole pine at all. And those populations will probably be extirpated over time, or sort of population extinction will happen over time.
Is it similar to what you see in your bats and the fruit flies?
Well, yeah, so under climate change, certain species will be favored, or certain populations will be favored. And so if with continued warming, the more tropical, warm adapted species may be adapting, or not adapting, expanding their ranges further north into the environments that are more conducive to them to be able to survive. Yeah. Yeah. So it will have broad ranging implications, of course. Yeah.
Another question from Eduardus Budries from Italy, I believe. He asks, I'm not sure who he's asking this to, but I think it could apply to any of you. He asks, is it possible to link the obtained SNP data, particular genes and their functions, and the functions that are related to climate adaptivity? Do you want to answer that, Aaron?
I can have a stab at it. So yeah, using these kind of genomic approaches in genome-wide association studies, you can find SNPs that are associated with certain traits that you might be interested in. And those might be in genes that are interesting candidates that could then be followed up. And depending on the system, you can do genetic manipulations where you knock out expression of the gene and test whether that gives you kind of the functional consequences that you might expect if that gene was playing an active role in adaptation to climate. So yeah, definitely, depending on the system and the data at hand, you can work towards that for sure.
Cool.
I can add something to that, it depends what organisms you work with. So if you work with the non-model organisms, especially if you work with vertebrates of species of conservation concern, a lot of these knockout experiments is not something that you're going to really do. And especially with long-lived organisms, a lot of the time, experimental manipulation is not really an option. So in these cases, we do really rely on just using, for example, transcriptomic data, seeing whether we see any changes in gene functions and trying to relate it to gene ontology. However, we never really have that direct causal link. We more have a correlative link. And this is really a weakness of systems that you work with. However, in terms of when we think about biodiversity, conservation, and the climate change, a lot of the species that are at the top conservation priority for many people are not necessarily these species that we're able to do these experimental manipulations on. So what we have to do is learn from experiments that were carried out on other organisms and perhaps try and extrapolate, rather than being able to directly link the genomic data to functionality.
I could comment on that if you'd like, Andrea. For the with our analyses of trees, we know what genes the SNPs, our candidate SNPs from genotype environment associations or genome-wide association studies with traits sit in. And some of those genes we can find annotations for from Arabidopsis or other plant species. But because conifers, they're gymnosperms, they diverged from angiosperms hundreds of millions of years ago. And so we have many genes that we don't have good annotations for. But I think something that we've found with our work is that you don't need to know to infer the implications for populations and for conservation. You don't need to know what those genes are or what they do necessarily. And sometimes we don't even need to have genes that we know are associated with traits or climate. We can work on overall genomic patterns.
Well, I think we've got time for one final question. We've got two questions here which are quite closely related from Zoe Diaz Martin and Shenka Malik. Both of them have asked about the relationship between geographic range size and ability to adapt to climate change. So both worded slightly differently, but very similar questions. Sally, do you want to comment on? I'll get you all to comment on that as a final point.
Yeah. So this is a question we're trying to ask right now. We are looking at one more narrowly distributed species, Western larch, that doesn't show much local adaptation to climate at all. And we're looking at a relatively closely related species, which is Douglas fir, which has a very large range and shows more local adaptation. Certainly species with wide ranges, and I think Orly has shown some of this, but with wider ranges, if there's gene flow among those populations, then warm adapted alleles can move more easily to populations inhabiting colder environments, for example.
Orly, do you want to add anything to that?
Yeah, just to say that indeed that's the case. Then one of the issues with endemic species, and especially those that are restricted to very specific conditions, is that the response to climate change will depend on, first of all, their ability to move away from these conditions if these conditions change, which is not always the case if we're talking about island populations. But the second thing is also the population size itself and the genetic variation that's actually available in the population. And in some cases, this may be quite limited population sizes as a result of the restricted geographic range. So in these cases, we may expect these species to be more sensitive to future changes and of highest conservation priority.
And Aaron, you work on very widespread invasive populations. Do you see something similar in your populations as well?
Yeah, we've estimated thermal performance curves for congeneric species, so other species of Zaprionis, I think 13 additional species, non-invasive ones. And we actually found that overall patterns of genetic diversity, levels of genetic diversity, are correlated with that thermal niche breadth parameter, which is probably correlated with distribution, at least anecdotally. Some of these island specialist drosophilids that are only found on one island, they have low levels of genetic diversity. When we estimate their thermal performance curve, they have a very narrow thermal performance curve. So yes, those species would be more susceptible to climate change for sure, and might be targets of conservation priority if you would like to.
Well, with that, I think we've come to it's now, we've hit the hour. So I think we've come to the end of our session. I'd like to say thank you again to all of our speakers, Sally Aitken, Aaron Comeault, and Orly Razgour, for their really good, fantastic presentations, really engaging. And we're also very grateful to Illumina, our sponsor, who made this webinar possible. We'd also like to thank you, the audience, for tuning in today to learn more about the role of genomics for biodiversity conservation in a changing climate. If you missed anything during this webinar or would like to listen to it again, our recording will be available shortly on the cell.com website. If you have any comments about the webinar or suggestions for future topics, we'd love to hear from you by email at cpwebinars@cell.com. Thank you very much.